Hello, fellow future biologists. In this genetic engineering sharing session, let's talk about CARs, or more scientifically known as plasmids. This model that we are showing is a recombinant plasmid. The green portion is the original plasmid before transformation. This bacterial plasmid will undergo gene splicing, which is where specific genes or gene sequences are inserted into the genome of a different organism. Who is responsible for splicing these genes? Well, this is where restriction enzyme comes into play. This particular enzyme will cleave at certain restriction sites, in this case, at the sticky ends with the sequence of GCTAA, right here. From this model, we will talk about how plasmids are biological CARs, the car parts, and the role of each part. This part acts as the car engine. It will determine which host the plasmid may go in, like how engines are made specific for each car model. Next, this part initiates a particular gene expression that acts as the steering wheel of the car to ensure that this process moves in the right direction. Then, we have RBS, that is a molecule that binds to a ribosome to position the message correctly for the initiation of translation. With every car, there has to be a door, so this MCS right here does just that. It opens the door that allows for the insertion of foreign DNA without disrupting the rest of the plasmid. When we genetically engineer this plasmid, a gene of interest will be inserted into the plasmid to form a recombinant DNA plasmid, which are the car passengers, inserted in between the MCS at restriction sites using restriction enzymes as mentioned before. Finally, we have the plate numbers of our car that differentiate it during the isolation process, which is markers such as ampicillin resistance and more. These components in our model are important to ensure our recombinant plasmid will work efficiently for preferred functions that resembles a bacterial plasmid. Now, when we talk about recombinant plasmids, restriction enzymes are mostly compared to scissors for cutting paper, but how similar are they? Firstly, blunt ends are DNA fragments being cut in a straight vertical direction, which is similar to scissors, but sticky ends, on the other hand, produce uneven and overlapping ends that is mainly used for recombinant DNA due to easy ligation with gene of interest. Moving on, let's identify our blunt ends that is cut by a restriction enzyme known as the SMA1 at restriction site. Genetic engineers tend to avoid blunt ends due to the ligation process being more inefficient by 10 to 100 times. How so? Because there are no single-stranded overhangs to hold the DNA molecules in position. When in between the CG is cut, the hydrogen holding the bases between the complementary nucleotide overhangs are also broken.